Okay, let's talk about meiosis today. And I want to kind of talk a little bit about chromosomes because it's pretty important in the development of meiosis and how meiosis occurs. So we have something called a diploid cell. Diploid cells are the cells that have a set of homologous chromosomes. And something that's homologous is where we see that each of the chromosomes from a male parent has a corresponding chromosomes from a female parent. So I have genetic information locked in a chromosome from mom and from dad, and when you put them together in the pairing that's seen, we call those homologous chromosomes because the genetic information is going to encode for certain genes, but they're pairing together. And so, we have 23 sets of homologous chromosomes that make up 46 chromosomes total, and each one of those chromosomes are pairing together for mom and dad, and they have the same, they're coding for the same genetic information. And so we represent diploid cells as 2n, and n is the number of chromosomes. And so for humans, right, we have 23, which is our n. And so if I'm multiplying this by 2, that's where I get the 46. So we contain 46 chromosomes. Okay? And so something that is haploid is where we only see a single set of chromosomes. So we're only going to look at one set of genes, one set of genetic information that's coming from one parent. And we know that the only cells that are haploid are what we call gametes. And so gametes are going to be our sperm and our egg. And we can represent a haploid cell just by the letter N. So for us, our haploid cells, whether it's the sperm or the egg, they only contain 23 sets of chromosomes. Let's just really quickly review mitosis. So we know with mitosis, we are looking at diploid parent cells. So typically we're looking at the asexual reproduction, and this will occur in somatic cells. Somatic cells we haven't really discussed yet, but somatic is dealing with pretty much any cell that's not, any cell that's not a sex cell. So nothing that is sperm or egg. So it could be a body cell. Really, soma is referring to body. So any cell except for the sex cells. And in mitosis, we actually do get two cells, so you could see in this picture, two whole cells that are considered diploid. So when you have cells dividing, multiplying and dividing inside of your body, to replenish maybe the dead skin cells that are on the surface. Any sort of division that's occurring there is going to be through diploids. So it's always 46 chromosomes that are getting placed inside. But when you look at sperm and egg, it's different. And then lastly, the daughter cells are identical to the parent cells because we're not going to see any sort of genetic information getting changed throughout this. It's just, multiplying it, replicating it, replicating, dividing, no exchange of genetic information. So in meiosis, just to remind you, we are talking about the creation of sperm and egg, where each of these represents only haploid, right? So that is why when you put these together, okay, you get something that is diploid. So just to plainly put it, meiosis is a process in which the number of chromosomes per cell is going to be cut in half through the separation of homologous chromosomes in a diploid cell. So we start off with a diploid cell and we bring it down to a haploid cell. We will see 2 rounds of meiosis occur, which is how we go from diploid to haploid. And the diploid cell, we actually end up getting 4 haploid cells. So we can make several sex cells just from one diploid cell. So let's just look at the development of sperm. We call this spermatogonium. And so we're going from something that has 46, this is our diploid cell. Interphase will give us some replication of the cell, and then we have sister chromatids that begin to show up, but we need to split this off and make them haploid. So we will see the development of the haploid cells, but then these are going to further break off to get us our 4 different cells. And those are all haploid. So let's start off with interphase number 1. Interphase 1 is where we begin to see the chromosome replication. So we actually see DNA replicate itself into 46 chromosomes. So we're at our 2N at this point. And then you move into our cell division. So it's very similar to mitosis. There's just little differences that you need to be aware of. So with prophase 1, the cell begins to divide, the chromosomes are going to pair up, but instead what we're seeing is the chromosomes actually pair up in what is called a tetrad. Okay, so we're looking at 4 chromatids, right, with their chromosomes, but 4 of them together paired up. And these are dealing with the same genes. So this could be mom, and that might come from dad's genetic information. And then what happens, this is what is really essential to the genetic diversity, right? If you look at your brothers and sisters, you're not genetically identical, and you're not genetically identical to your mom or dad. And it's due to this portion, this thing that right here called crossing over during prophase 1 of meiosis. And so what we see is those tetrads, they've come together, and what they do is they actually swap some genetic information. They literally exchange those homologous chromosomes, cross over. That's why it's called crossing over, and we get that variation, right? Because now, look, this genetic information right here is going to be different. So this region maybe codes for eye color, or the shape of your nose. And because of this crossing over, the shape of my nose looks very different from the shape of my brother's nose, right? I'm not as tall as my brother. My brother is taller than me. And it's because of this genetic variation, this crossing over. Okay, so in Metaphase 2, it's basically looking just like Metaphase from mitosis. But instead of splitting up the sister chromatids, we end up actually lining up those tetrads. So the tetrads still line up with the centromeres along the middle of the cell. The spindles come, the spindle fibers come and get ready to attach to those tetrads. And then in anaphase, that's when we see the splitting up of those homologous chromosomes toward the ends of the cell. So again, we haven't split sister chromatids yet. We just split the tetrads. So we have whole homologous chromosomes. That is homologous chromosome, that is homologous chromosome. So they are splitting apart. And notice, right, if you see that little red, okay, the genetic information has been created, so we can still have our variation. And at the completion of this, the separated chromosomes are going to cluster at the opposite ends of the cell, very similar to mitosis. And then we still have our telophase occur, right? You got the nuclear membrane that begins to form. You have to reform the cell, and then cytokinesis is that splitting of the cytoplasm, so we can have 2 cells. So that was meiosis 1. Okay, now we need to get down to the single 4 different sperm or egg cells. So in we'll take a look at meiosis 2. And so remember, in meiosis 1, 2 cells are made, but they're genetically a little bit different. That's really the reason for meiosis 1, is that we want to get some genetic variations. We could put that into each individual sex cell. In meiosis 2, the cells do not undergo a round of the replication before entering. So now we're going to take it and further split those chromatids apart. So there's really no interphase during this time. So prophase 2, now it starts to look pretty much just like mitosis. The chromosomes that have the chromatids become invisible. Sorry, battery's almost dead. I better hurry. Metaphase 2, the chromosomes line up at the center of the cell, just like in mitosis. And then those chromatids are going to attach to the spindle fibers, and then the paired chromatids will separate during anaphase, right? We see the spindle fibers kind of pull back on those chromatids. And then telophase 2 and cytokinesis, it's the very same thing that we've seen in mitosis. And what we get now is that each of the 4 daughter cells have 2 chromatids inside for this example. And look, if you look at the picture, you see that genetic variation. This one has all yellow. Okay, this one right here has some red and yellow. This one has more red. This one has a little bit more red, but all very genetically different. Okay, and these are all haploid cells now. So in this case, there's only 2 chromosomes instead of 4. And that's how we get our 4 different single cells. Okay, so let's compare the two. With mitosis, we have 2 sets, pretty much just 2 sets of chromosomes, and we have meiosis, we only have 1 set of chromosomes in the end. We have diploid cells at the end of mitosis, and in meiosis we have a total of 4 haploid cells after 2 rounds of meiosis. Mitosis, we are dealing with autosomal, or another name is somatic, chromosomes, and in meiosis we are looking at sex chromosomes. So we're looking at things that are for the sperm and the egg. In mitosis we are making an exact copy, and in meiosis we are looking at genetic variation that comes from crossing over. In mitosis we get 1 round of cell division, but we have to have 2 rounds of cell division in meiosis to complete the cell division. And that's it.